Welcome to DNA Replication Nation, where we can learn about DNA replication and soar to new heights.
Oh, hey, Dr. Charles. We were just talking about DNA.
Yeah, we know that DNA gives the body genetic instructions for the development and function of every part of the body.
And it is made of 4 nitrogenous bases. They are adenine, or A, thymine, or T, guanine, or G, and what was it?
Cytosine, or C.
Oh, thanks, Mimi. You really got my back.
Or your sugar phosphate backbone.
Good one.
But really, this is no laughing matter. The sugar phosphate backbone is very important because it is the major structural component in DNA molecules.
And DNA molecules are very important. But one thing that Eugene and I don't understand is how DNA replicates.
Well, I know that it occurs in the nucleus during the S phase of interphase in the process mitosis.
That's the process through which a cell divides. But how exactly does the DNA replicate?
Well, Mimi, it's a semi-conservative process where 2 parent strands of DNA separate in order to create 2 daughter strands of DNA. The replication occurs in the 5' to 3' direction.
How does it begin?
Helicase.
Oh.
It is an enzyme that unzips the helix at the replication forks, like a zipper.
Right. Then the RNA primase directs where the RNA primer should start in the 3' to 5' strand, or the lagging strand.
Is that when the DNA polymerase 3 allows the addition of free-floating nucleotides on the 5' to 3' strand?
Yes.
Yes, and then the DNA polymerase 3 also starts at the primase on the 3' to 5' strand and fills in the space between, or the Okazaki fragments, with free-floating nucleotides.
Good. Now, what can you tell me about DNA polymerase 1?
Isn't that what we were just talking about?
No! But seriously, the DNA polymerase 1 removes the primer from the 5' end and replaces it with DNA nucleotides.
Just like they would take your plate at a restaurant and replace it with a dessert.
Sure. Then DNA ligase would come through and attach the sugar phosphate backbones to the nitrogenous bases. This is the parent strand, and this is the daughter strand.
And that forms 2 identical strands of DNA!
Yes!
Yay!
Sweet! Hey guys, how do you eat DNA spaghetti?
With a replication fork!
Yeah. Whoa! Earthquake!
Oh my gosh!
Whoa! Typhoon!
Okay.
I'm afraid I'm gonna drift too far.
In the 5 to 3 direction. Put down.
It began. You actually said it too. Hey, Dr. Childs. How does it begin?
Of every body, every part of the body.
Or nutrigenous bases. Oh no. Oh no. Oh no. No, it's not, Star. Oh gosh.
Keep going, just let it happen.
Well, I know that it.
This just made my week worthwhile.
No, don't do it.
And your feet will be soaking too. Good thing I'm wearing high waters. This water might get high. Oh my gosh. Oops. Should I step on it?
No, you'll fall in.
Oh gosh. My shoe's all wet. I have a hole in my shoe. Gross. Look.
Well, Mimi, it's a
No!
Yes. Number 1. D-R-C rival.
Where's my agent?
Stupid DNA.
I don't know.